to my channel. I'm Tapushi Konkar and it is my channel Pure Biology. And before going to the topic, I want to say thank you to all of you who have subscribed my channel and give me support to grow my channel. Okay. So today's topic is actually the Lac Operon, and this is the second part of the Lac Operon, you can say, because before that, today I explain about the positive regulation and the negative regulation, and it's a previous video. In previous video, I already have explained the parts of the Lac Operon, what are related or what the parts of Lac Operon, that means the promoter and repressor and operator. I totally have explained about that in the previous video. So you just first check out that one, and after that follow this one. So you just follow the I buttons that generally I give in my topic or in my video. You just follow that one, or sometimes I give the link in the description box, or you can follow the playlist also, because I try to actually attach the related videos with the particular video. So you can obviously check out that if you follow the I buttons, or you can check out from the playlist. Sometimes I also try to give the link, video link in the description box, but it's very rare. Generally I place the link in forming the playlist or in forming the I buttons, so you can obviously easily get the linked video from the I button. Okay, so let's start. So what is the operon? I already discussed about that. So today's topic is actually the positive regulation and the negative regulation. So first I briefly say that this is the I means first these are the structural genes, and these are all the enzymes related to these structural genes, and also another part of the Lac Operon. These are the I, P and O, promoter. This is the repressor gene, and this is promoter, and this is operator. Okay. So what happened in case of positive regulation? Generally, the products of the regulator gene. What is the regulator gene? This is the I gene. Okay, this is the regulator gene. This acts as an activator or the repressor. Okay, as it helps to activate the transcription or the translation process, or sometimes it represses the total procedure. Okay, so the binding of an activator protein to its target DNA site and increases the rate of transcription. Okay, so in that case, the protein that is produced by the regulated gene, that actually known as the activator, because in presence of this activator protein, it is very helpful to continue the process, and I will discuss about this in my another video, but today I just briefly explain it. So its presence is very important for continuing the total process, and that's why the protein is called the activator. Okay, protein is called the activator, and generally it binds to the target DNA site. Okay, target DNA site to increase the transcription process, and it is known as the positive regulation. So you can obviously understand why it is the positive regulation, because its presence is not effective for continuing the process. Okay, so that's why it is activator. That is this protein that is formed from the regulated gene. So these are very important, and these are helpful for this process. So these are called the activator, and the total process is called the positive regulation. Okay, this is called the positive regulation. And second one is the negative regulation. What is that? Just opposite of the positive regulation. Okay, just opposite. What happened in here? In absence of inducer. Okay, in absence, when the inducer is not present. Okay, inducer is not present. What happened in that case? The Lac repressor that binds to the operator site. Okay, suppose This is the repressor protein. You can see this is the repressor protein, and that binds to the operator site just between the promoter and the Z structural gene. Okay, and P is the site of the RNA pol. Okay, RNA polymerase. So as the regulator gene, this one the regulator gene that produce one kind of protein. This is called the repressor protein. It binds to the operator. Okay. So what happened in that situation? It prevents the transcription. Okay, it prevents the transcription. Because I explained it, the transcription process of this gene is totally stopped. Okay, and here the progression of the RNA polymerase transcription is blocked. Why the transcription process is blocked? Because the RNA pol that binds to the promoter site. Okay, so when and it generally helps in the transcription. So the blocking of the RNA pol or RNA polymerase is totally occurred due to the binding of the regulator protein to the operator site. I think you can understand. Okay. So what happened if inducer absent? Clearly understand. Inducer absent. Here the inducer absent. Inducer if absent, that means the regulator protein that binds to the operator and block the RNA polymerase its progression. And as usually the progression of the RNA polymerase is blocked. That's why the Z, Y and A, these structural genes that cannot transcribed. Clear? Cannot be transcribed. And so translation also be stopped. So here this is called the negative regulation. Okay, and the repressor protein, that is the Lac repressor protein, is called the negative regulator. Okay, this is called the negative regulator, the Lac repressor protein. So here this is regulator protein, negative regulator. Clear? But what happened? And the structural gene also repressed here. And when the inducer is present, okay, this situation occurred absence of inducer. And when the inducer is present, in that case what happened? Suppose inducer is not there. Okay, so what happened? Inducer is present. That means inducer is in the medium. So it binds to the allosteric site of the repression. Okay, if the inducer present, okay, that bind to each allosteric site. Okay, allosteric site. Where it binds? The allosteric site of the repressor protein. The inducer binds with the allosteric site of the repressor protein. Clear? And inactivating the operator binding site of repressor protein, and it cancel the binding of suppose this cannot bind. Okay, so binding is not possible, and okay, this is it. So binding is not possible, and what happened in that situation? Formation of allolactose is catalyzed by the beta galactosidase. This is another thing. So what happened? As the repressor, inducer just binds to the allosteric site of the repressor protein. The repressor protein cannot bind with the operator site. Okay, so that's why the RNA pol that present on the promoter, it cannot be blocked. Okay, it cannot be blocked. So the transcription of the Z, Y and A structural gene is possible, and also possible the translation. Structural genes transcription is possible, and after that translation also be possible. Okay, and here one thing is very important. The lactose is not very important here. The important thing is the allolactose. Okay, you just remind it, or you can just keep it in your mind that allolactose, because Lac Operon, so we can say this is the lactose, the inducer. No, lactose actually cannot help in the binding. Okay, here very important, that is the allolactose. Okay, this allolactose is very important, and this is actual inducer. Okay, this is actual inducer. So formation of allolactose, that is catalyzed by the beta galactosidase. Clear? This beta galactosidase enzyme that bind to the Z structural gene, or that is very important for the Z structural gene. So this enzyme is responsible for the formation of the allolactose. Clear? This is formation of the allolactose by the beta galactosidase. Okay. So what is the function of here the lactose? Lactose is capable of inducing the operon system. Okay. It just only help to induce the operon system, not the serving as a substrate. Okay. So what is the substrate? The substrate is the allolactose. Clear? The substrate is the allolactose. Lactose is only inducer, and allolactose, that is actual the substrate. Okay, that helps to bind with the repression protein and block the binding of repression protein to the operator site. And as usually, the RNA poly proceed, and it helps the transcription of the structural genes. Clear? And here the lactose is capable of inducing the operon system, and so it is called the gratuitous inducer. It is called gratuitous inducer. Okay, and one example of the gratuitous inducer, that is called the IPTG. Okay, this is called the IPTG, and full form is the isopropyl thiogalactoside. Isopropyl thiogalactoside. Okay, this is IPTG. And so what is called the lactose? Lactose is called the gratuitous inducer. So this is the positive regulation and the negative regulation of the Lac Operon. Okay, another term also I have to explain you. First I rub it and then I will explain. Okay. What is called the coordinate induction? This is actually occurred in the presence of lactose and in absence of glucose. Here, presence of lactose and in absence of glucose. This situation is generally occurred. Okay. So the amount here, what happened actually here? The amount of each enzyme that increases coordinately about 1000-fold. Okay, so the activity of the enzymes, all the enzymes, that will be increased to 1000-fold. Okay, and in that situation, what this situation arise? So what happened actually? Absence of glucose and presence of lactose, the inactive genes that are now actively transcribed. Okay. Absence of glucose and the presence of lactose, the structural genes that are inactive, that remain in inactive situation, that becomes actively transcribed. Okay, so this situation is called the coordinate induction. Okay, so what happened in coordinate induction? Actually, the enzyme's activity that will be increased to the 1000-fold, and as a result, the genes activity that were actually inactive sometimes. So the activity of these genes becomes rises. Okay, so in that situation, this induction or this situation is known as the coordinate induction. Okay. Actually, the activity is increased, and the transcription process of the structural genes also be increased. Okay, so this has happened actually presence of lactose and absence of glucose, and due to the enzyme's high activity. genes transcription also be increased. Okay, this situation is the coordinate induction. And this is the Lac Operon's negative and positive control. I explained positive control. I explained you elaborately in another video. Okay, today I explain just briefly what is the positive regulation and what is the negative regulation and what is the coordinate induction. Okay, so this is the second part of, you can say, the Lac Operon. And in the next part I will describe about the mutation. What kind of mutation we can see in the Lac Operon, or due to the mutation of the genes, what situation may be arised. So these are described obviously in the next video. Okay.